So I hereby open this academic session in which Yawen Wang will defend their PhD thesis entitled Challenges and Potentials of 7T fMRI for Investigating Attention and Perception. The candidate has now 15 minutes to expose her thesis, and then we will ask some questions. The floor is yours.
Highly esteemed prorector, dear members of Corona, supervisors, colleagues, friends, and family. It's my pleasure to present to you the research I have done during my PhD thesis, titled Challenges and Potential of 7T fMRI for Investigating Attention and Perception. Magnetic resonance imaging, MRI, is a non-invasive imaging tool that has been widely used in healthcare and research. By using powerful magnet and water molecules inside our body, it generates detailed images that shows different tissues. It can also be used to measure brain activities when a participant engages in a cognitive neurotask, as it's called functional MRI, or fMRI. Moving to ultra-high field, such as 7 Tesla, there are many advantages. The most common one is to obtain higher resolution images with more details to see a smaller structure. We can also acquire fine-grained functional activities. With this, we can even probe brain functions within areas at layer or columnar level. Holding those promises of studying brain functions at ultra-high field, in here, we explore the neuromechanism of attention and visual perception. We challenged ourselves to study a region that is crucial in attention, but has also been difficult to image. It is nucleus bezalis of Maynard, the NBM. It's small in the middle of the brain. It produces a neurotransmitter called acetylcholine for all parts of the brain, as you can see by different colors. Why are we interested in this region? Because it participates in attentional process. Let me explain with an example. On a beautiful morning, 3 mountain goats are enjoying their life. But little do they know, their happiness hinges on their ability to spot a hidden leopard. Their task requires keen focus. They might need to pay attention to the sound or the scent from the environment to detect the leopard. These two types of attention tasks might involve in different parts of the brain, the NBM. Therefore, our initial plan was to use 7T fMRI to see if an auditory attention task can activate different parts of the NBM compared to a visual attention task. This was quite ambitious, but we are encouraged by the existing body of research indicating measurable changes in the size of NBM, even with 3T MRI. When we collected our own MRI dataset, we saw that even on our higher resolution anatomical image, we cannot see the NBM. We need an atlas to localize it. But the atlas is at lower resolution, and we know the NBM showed in red here cannot be this big. Besides, there are some issues on the resampling as well. So we want to investigate how best to localize the NBM, but also understand its structure. After reviewing 37 neuroimaging papers, in this pie chart, we show that only 26% of studies report the NBM volume with a meaningful physics units. Even within this 26%, we notice a wild variation in the reported volume estimates. For example, the difference of the NBM volume estimates from Taipei et al. 2016 and Marcello et al. 2018 are so huge. Both these studies cannot be accurate, so we decided to dig a little bit deeper. The majority of the studies we have been reviewed used an atlas to define the ROI. We also see variations among those atlases. As you can see on the figure, using same threshold, different versions of atlas give a different estimates. On the right figure, by taking one atlas, changing the threshold, you can also see a wild change of the volume estimates. Using a threshold at 0.3 and 0.6, the volume difference is also huge. Because of this, we don't know which threshold is the best to pick. Therefore, we developed our own mask as a ground truth using big brain dataset. We show that lower resolution leads to unreliable results, and a higher resolution of 1.2 millimeter isotropic or even sub-millimeter resolution are recommended. However, we still did not solve the problem of being able to see the NBM on our anatomical dataset. We narrowed down this issue to be one of the MR contrast, and we illustrate this by being able to delineate using a quantitative T2 star image, shown here where the NBM is bright. Although the image was required at 9.4 Tesla, but it is a post-mortem dataset. I hope with the advancement in technology, such as the 14 Tesla MR scan in the Dundas, it can facilitate the future investigations on the anatomy and function of the NBM inside the living body. So here we have a look at the region that is very important in attentional processing. For the next study in this thesis, we probe another important aspect of our perception, which is brightness induction. For this turn, we turn our attention to the visual cortex, as it's shown here in red. There is currently a debate in the field whether the visual cortex is involved in the process of brightness induction. If it does, is it in V1 or in V2 or in even higher order area? At this point, you might wonder, what is brightness induction? Here is an example. You might think the two small squares are different, but they are actually the same. The different perception is induced by its background. In the rest of the representation, the background region where change is luminance will be referred as inducer, and the region where we perceive the different perception will be referred as probing region. To put it simply, in this illusion, the luminance change has an anti-phase relation with the change of our perception. A study from our own group using 3T fMRI reported an anti-phase relations in V2 using an annulus stimulus, as you can see in the highlighted area. Encouraged by these results, we went to higher strengths of 7 Tesla and a higher resolution of 0.8 isotropic. We also adapted our stimulus for the 70 MR scanner and precisely delineate our inducer and probing region. This is how it looks like in the brain. For the cortical region representing inducers, we expect to see a strong positive fMRI response and a strong negative fMRI response when the luminance decreases. For the cortical areas representing probing region, we expect an opposite trend. However, in our own data set, we only see a strong negative fMRI response when the luminance decreases. Increase in luminance did not induce a noticeable cortical response. In the probing region, both conditions did not induce a very successful cortical response. Our findings can suggest that the early visual cortex may not play a role in this process. Well, there could be various reasons for this, but one thing we can control is how we design our stimuli. We need a better stimuli that can induce a balanced cortical response towards the two conditions. Meanwhile, we can minimize the mean luminance difference between the two conditions. and more importantly, we want to create a stronger brightness illusion. This leads to the final chapter of my work, where we study Anderson and Wiener illusion. It is a more powerful illusion than the one we used before. Believe or not, the circle on the left are the same color as the one on the right. To destroy this illusion, we only need to rotate the background by 90 degrees. This makes it an ideal control condition for brightness illusion. Inspired by this illusion, we made our own implementation of the illusion by changing the luminance over the full background, as shown in the blue line in A. We also found that we could generate a similar illusion by changing luminance only close to the probing region edge, as shown in the blue line in B. Using this novel technique, we found we could generate very striking illusions, which the brightness of the probe varied dramatically, while the manipulation of the background was difficult for participants to be aware of. Using this technique, it largely reduced the mean luminance difference between the two luminance conditions. For comparison, in the original illusion, it is very clear that the background is different between bright and dark probes. To quantitatively test the effectiveness of the stimulus, we ask the participant to compare which probe in A or B appears brighter than C, because this is where we really change the luminance of the probe. Because this project started at the beginning of the pandemic, so we collected the data through online platform. As you can see, our new stimulus A and B induced brightness illusion successfully, but the rotation only reduced the illusion rather than destroy it. So our exploration shows that this set of stimuli works only partially on an online platform, and a re-evaluation in a psychophysics lab is needed. To summarize, we have addressed the issue of accurate measurement of the NBM, a very important structure for attention, in Chapter 2. In Chapter 3, we explored the role of visual cortex in brightness induction using 70 fMRI. In Chapter 4, we developed a novel brightness illusion stimulus using online psychophysics. Although our hypothesis has not been fully proved along the way, I believe it paves the way for the future studies on similar topics. With this, I would like to thank for your attention, and I would like to give the word back to the prorector.
Yes, dear candidate, thank you very much for this very clear exposition of your thesis and for being well on time. Of course, we will now start questions session. It's a weird situation because I'm both the prorector, but I'm also going to start the questions, so I will introduce myself. I am Federico De Martino. I'm professor at Maastricht University of Sensory Circuits and Neuroimaging. And yes, I will also ask you a question at this point. And as it's custom, I will start with congratulating you and your supervisory team with this work. I read the thesis already some time ago. I reread it more recently, and I found it inspiring. I think that from this is the way we learn, that finding things that don't work are actually the thing that inspired me the most, solving problems, figuring out how to make them work, and moving a step forward. I hope you surely did not show any discouragement during this work, and I hope you are proud of where you have come. So your supervisors and co-supervisors should be proud of the work you all have done together. Now, of course, I liked your thesis a lot, but I am forced to have to ask you some questions. So I will ask some questions. My first question is on chapter 2, where you are trying to delineate the nucleus basalis of Meinert, and of course I'm attracted to this chapter because originally I wanted to do this work as well with you, and of course I realized very soon that it was challenging, and I'm very proud to see where you've brought it together with the others. And I think it's really nice that you went through all this thing of looking at an atlas and looking at things, but then also bringing it to back to MRI with this post-mortem data set that was collected at 9.40, and actually showing that if you have the right contrast, if you have enough signal, if you have the right resolution, in the end, you can actually see it. And I think this is great. It really is inspiring. But I know a little bit about post-mortem MR, and I know that this data set that you used took very long to collect. So post-mortem MRI data sets like that, they can take up to a weekend of scanning to collect. And they have a big advantage, that is that the brain is definitely not moving because it's post-mortem. So although you say that you hope that with bigger scanners and everything, things will happen in vivo, I'm actually curious to understand how you think this will happen, because, or what at least you think would be the biggest hurdle to bring this approach to in vivo scanning. So what is, you think, the biggest problem to try and do the same thing you've done now post-mortem, but in vivo?
Highly esteemed opponent, thanks for your compliments and question. And I admit that the post-mortem data are very different from in vivo data. And but one thing with moving to ultra high field or even higher strength, we might have better contrast, or we might can acquire higher resolution. But it cannot directly transfer what we find from post-mortem data to the in vivo data set. Like the T2 star value from post-mortem will be also different from the in vivo data set. I think if we want to do this project again in vivo, I wouldn't want to do the function first. I would want to do the anatomy. And because it has been shown that different part of the NBM connect to different part of the cortex. And I think it would also be very great to show, like in our anatomical data, this is also true. We can show it in MRI data set. I think also like when you move into 9.4 or even higher, and the bore of the scanner is also become smaller, it become more uncomfortable for participants to stay still and lie there very long. So I think we also need to figure out a way to how to make those second session to be more comfortable and to more doable for participants, for allow us to acquire better quality of data set.
Okay, then there is definitely work to be done, and that's nice. If I may, I will ask one more question before I give the word to the others, and I'm sorry that I do so, but I'm going to make a jump, so I'm going to jump to a different chapter, and I'm going to go to the brightness induction chapter. I mean, it's a very interesting study overall, and it's inspired a lot of thoughts in my head. I do a little bit of a laminar fMRI as well, or high-resolution fMRI as well. But there is one thing that I found interesting in the results that you don't discuss a lot. And that is the fact that if you look at, so you also analyze the effects in the edge of the probing region, not just in the probing region and in the inducer. And in some way, if you look at the results comparing the inducer to the edge instead of the inducer to the probing region, the results look better. In a sense, you still have the problem that the brightness increase doesn't work well, but for the brightness decrease, the effect is stronger. So why is this the case? So why is it the case that the effect is so strong on this edge representation of the probing region compared to the probing region itself?
Thanks for your questions. And I would think because of the edge has more contrast compared to the very center of the probing region, and also most of the, as you can see, so the strongest fMRI response is driven by the edge response, and also by connecting this question with later the experiment in chapter 4, and we also show that the edge is actually very important to induce brightness illusion. And in the chapter 4, we only change the very narrow band of the edge close to the probing region. We already can induce very strong brightness illusion. So I think the edge part does play a very important role to generate this illusion.
So, and as a short follow-up then, does this mean that if you were to try to do this again, you would change the stimulus? So you went for this very large probing region in the center, and from what I can see then, it means that maybe you have this whole big part of the field of view in the center where nothing is really happening, so, or at least not as strongly. Would you then change the stimulus and maybe go back to stimuli that have been used before, using more like annulus type of representations, or do you think that it's still a good idea to maximize the size of the probing region?
I think if I would restart the project, so I think because from our psychophysics we already in chapter 3, and we showed that size of the probing region or the inducers doesn't really make a big difference on the illusion, but we also keep constantly observe the illusion is unbalanced. Luminance, a decrease in luminance does make a stronger brightness illusion, and if I want to directly use this paradigm in the fMRI again, so I think I will do a staircase experiment inside the scanner to maximize the illusion in the scanner, because we observe the level of the gray surface really influence how big the illusion is. So I will maybe find a better gray square to make it balanced, and also at the same time maximize the illusion. But regarding what we found in chapter 4, because it's a very different one, because it also involves with low frequency stimuli. And even though the edge can induce the brightness illusion, but it is not as strong as when you use the full background. Okay. So this can mean, so when you use a more complex stimuli, there might be more mechanism involved. Even though if we use the same set, but only the stimulus is different, we might find something, but the mechanism behind these two sets could also be different.
Okay, thank you very much. Thanks for the answers. I think it's time for me to give word to the next opponent, and the next opponent will be online, and it's Professor Floris de Lange, Professor of Predictive Perception and Cognition from the Donders Institute for Brain Cognition and Behavior at Radboud University in Nijmegen. And I'll give the word to Professor de Lange.
Thank you, Professor. Oh, I hear a bit of an echo, but let's hope that that's not too disturbing. Dear candidates, dear Yawen, first of all, let me tell you, I'm sad I cannot be with you physically today, but I am very happy that I'll join you online. I really enjoyed reading your thesis. I thought it was really great work. It's very rigorous. It has a broad scope, I thought. It encompasses both psychophysics, high resolution fMRI. You did some outstanding anatomical work, and it's all very well couched in theoretical frameworks. So I learned a lot from reading your thesis. So congratulations to you for that, and also to your supervisory team, Professor De Weert, Roberts, and Kasia. You also included some propositions in your thesis, and I always enjoy that. So I wanted to start off with one of the more frivolous propositions, which I thought was funny, the number 9. So there you say, following B.F. Skinner, it would be more useful to scan one brain for 1,000 hours than to scan 1,000 brains for 1 hour. Now, one of the reasons I found this amusing is that I doubt that Professor Skinner would found it useful to scan the brain at all, given that he is a behaviorist, so he really doesn't care much for the brain, I think, or for internal representations. But if we go with it anyways, I'm just curious first, what brought you to this statement? Can you defend it for me? Why do you think it's more useful to scan one brain for 1,000 hours than 1,000 brains for 1 hour?
Esteemed opponent, thanks for your compliments and question. When I wrote this proposition, I was thinking, I think there are two goals of the research. One is to prove something exists or not exist, or another thing is to generalize the conclusion, whether we can do it to a broader population. I think for like an ultra high field study, we normally have a very small sample already. So I think it's more, if compared to the generalization, I would rather prefer to prove this effect is robust and it does exist in some brain. So I think by this, I would like to see if you scan one participant for more sessions will be more helpful than just scan multiple persons for one session.
Okay. Yeah, now I see where you're coming from then. But just to play devil's advocate, I mean, we know that there is appreciable variability in the population, of course. So you might be sort of unlucky to scan this one person that is not sensitive to the brightness illusion that you are interested in, or who has a V1 organization that doesn't allow you to actually study the phenomenon of interest. So in that sense, could you say that we want to have knowledge about how things work on average, rather than in the one specific person that you happen to study, for which indeed you might find things that then do not generalize, but it could also be the other way around. You might not find something that is actually there in the population for idiosyncratic reasons.
Yes, I think that will also kind of will happen. But I think even though we are very unlucky and then the participant is very special, and then we do study him like for 1,000 hours, but I think it's also very interesting to report this special case, and maybe also like to broaden our views on how the visual cortex looks like, and how maybe he has a different mechanism to process those kind of surface. And maybe I had a very improper example. It's like we think most of the males have XY chromosome, but it's not all the males has XY. It can be the Y that is not functional, but it can also be XYY. But we can still make the conclusion that most of the males has XY chromosome.
Yeah, no, I agree with you. And I think ultimately, whether to study 1 or 1,000 individuals may also depend on the question of interest, of course. So I think for some things, inter-individual variability will be more important than for others, intra-individual variability will be what you want to, what you care for most. So let me move to a sort of a less philosophical or more empirical question. So my question is about chapter 3. And there I was, my first question is a sort of a clarification question. So I was looking at figure 3.15, which is on page 116, where you give an example of the Bolt response for the two luminance conditions in the inducers. And I think I am misunderstanding something about this figure, because when I compare it to the figure before it, figure 3.14, I can clearly see that there is this kind of either no difference or like a negative Bolt curve for the inducer regions. But then in 3.15, it's in the legend, it says it's like it's the Bolt response in the inducers, but here it's positive in both cases. So I think I'm just misunderstanding figure 3.15. Can you help me understand it? So is there like a difference or a discrepancy here between figure 3.14 and 3.15, or what is 3.15 showing?
Thanks for your question. And figure 15 shows like a predicted Bolt response from FSL, shows how the most significant voxels, how the Bolt signal look like.
But is this then taken from an edge region, or is this taken from the inducer ROI?
I think it's from it's supposed to come from the edge region because it selected the most significant voxels towards the luminance change. I think it comes from edge.
Oh, okay, got it. Yeah, now then I understand. Now in the figure legend it says in the inducer, so I thought you meant the inducer ROIs, but it's so then I understand it's the edge ROI. Yeah, no, so I had the more general question I had with this, and I think you actually address it a little bit in your discussion, is like, what would you do differently if you could do this again? Because there's in chapter 4 you use a different type of stimuli that is more powerful. So maybe I'll ask a quick question on this, because then I also need to give the floor back, I think, to the pro-rector. So do you think you could use the stimuli that you used in chapter 4 also in a 7T experiment? And if so, can you then cleanly delineate the same regions that you have, like an inducer region, a center region, when you would use such stimuli? Or would this in the end be problematic?
I think this one thing I mentioned that because we done these experiments online, so the re-evaluation is also needed. And as far as I know, some students from my lab, they already test the illusion in the lab, so they do observe a stronger illusion in the lab, but unfortunately they did not test the rotation effects. So I think first I would like to test whether the rotation will really destroy the illusion at all. And if it does, it will make a perfect fMRI stimuli, because it has very small luminance difference between the luminance increase and the decrease, and it will also reduce the effect of when you change the whole luminance background, you might also get some reflections, and especially to increase the luminance to white. And it also have little bit influence to the fMRI response. So if we can confirm that the rotation is very effective, I think we can move this into fMRI.
Okay, thank you very much. I'm fully satisfied, and I give the word back to the pro-rector.
Yes, and the opposition will now be continued by Professor Kamil Uludag, Professor of Medical Biophysics at the Medical Biophysics Department of the University of Toronto. And Professor Uludag is also online, so the word is to him.
Okay, dear candidate, it's good to see you again.
So it's a challenging work what you have done. So a lot of different techniques and different data and different analysis tools. So congratulate on that you managed to find your way through. So given the time, I will jump to my first question. So in the conclusion section, you write, in retrospect, it would have been better to first conduct the study with supramillimeter resolution at 3 and 7 Tesla, using the stimulus we planned to use. At 7 Tesla to first observe the main effect of interest. So you argue perhaps it would have been better to test first in a low resolution and then go to higher resolution later. However, given the data you already have, can you not test whether the spatial resolution is the problem in the failure of getting the effect you wanted?
I think there might be two things we can try. First, maybe we can try the smoothing while we process the functional data, because we didn't do any smoothing because we want to do layer FMI later. So we think, so if we just want to confirm the effect is there, or in way 2, we can try have a smooth kernel to see whether it will kind of boost the signal. And also, another thing, I'm not sure whether it's a good idea, so we can also downsample our data to the functional resolution we want, and then analyze the data set. We can see whether the effect is there.
Yeah.
I think that's right. So therefore, I think you can test with the data you have the issue of resolution and then see what happens. The second thing, what I was wondering, one of the main motivation of acquiring the high resolution was to do layer analysis, but you didn't do any layer analysis, and you justify it because you didn't see any main effect. Can you think of a scenario that you don't see any effect sort of globally, and there would be still layer-specific effects?
I think there might be some layer profiles, but I'm not sure how we can trust the data set if we see the effect. Even though our effect is not very significant, but we do see some trend, such as in the luminance decrease condition, we do see like the opposite trend. They have a very significant negative response in the probing region, and also a bit positive response as well. So we do see this antiphase relationship for the luminance decrease. I think with this cortical response, we might expect to see some level of layer FMI, but I think it would be better if we do see like a very confirmative response, and we know what it means, and then we can explain the data more better.
And I think we have some issues online. So in the interest of time, I think we can go back to Professor Udag when he comes back online, but we will continue now the opposition with Dr. Peters. From the Department of Cognitive Neuroscience at Maastricht University. The floor is yours.
Thank you. Dear candidate, I would like to join my colleagues in congratulating you on this very nice, rigorous work that you've done.
Please use the microphone.
Congratulations. Oh, sorry. Congratulations for the to the supervisory team. And yeah, as I mentioned, I thought that your work was indeed very rigorous, especially chapter 2. It's, I think, important work to indeed review more often work that is already done, and especially in this case, I was surprised by the differences that you found between the different studies. And it's so important for a target, especially if you want to, for example, consider this nucleus in, as I understand, clinical research targeting with deep brain stimulation, for example, in dementia. So I was wondering whether you have some suggestions, because if you want to use this for clinical research, you discussed with the pro-rector that it would be nice to use a 9.4 for increased field strength, and you focus a lot on that. But we don't have that many 9.4T scanners, of course, but we do have 70. So if you go back and would be able to do this study again, is there something that we can, at the 70, improve to better localize this nucleus?
Esteemed opponent, thanks for your compliments and question. I think, yes, indeed, we don't have 9.4 Tesla everywhere. And I think from our own observation, we also acquired some MP2 range. And for that, we have several kind of quantitative T1 from the MP2 range. So from there, we have the resolution of 0.65 millimeter isotropic. And we see a small bright region, but it's not very obvious to see, but it might be there. So I think with 7 Tesla, we can also try some of the quantitative imaging at the region. Maybe we also can see some structure over there.
Okay. Thank you. I think in the interest of time, I should skip my second question and give the word back to the pro-rector.
I think there is time, but maybe what we can do is you can ask if you have one more question, and then we can go back to Professor Uludag, who maybe wanted to ask a question after that. So if it's that okay, you can ask your second question now, and then we'll go back.
Okay, yeah. So then I would like to switch to chapter 4, or I have a question that's more general on the different stimuli that you use in chapter 3 and 4, because you mentioned several times that you want to increase or you have some ideas on maximizing the brightness percept. And I think I heard correctly, you didn't discuss this in your thesis, but that you tried several things. So for example, the size didn't have that much effect, but you see an effect of spatial frequency. I would have expected the opposite. So I'm curious what you think causes this spatial frequency effect, and why you don't think that there is an effect of size.
Thanks for your question. I think regarding to the size, effect of size, this was done in chapter 3. So it has a different, we use a uniform surface to induce the brightness. But in chapter 4, we have a cloud surface. And I think because from the pilot, we observed that frequency can have an effect when you increase the spatial frequency, and the illusion will become weaker. And this would mean that with lower frequency stimulus, there might be involved some neurons have larger receptive field, and then this might influence or interact how the person to segregate the figure of background. Mm-hmm. And I think this might be one reason why we can have a stronger illusion compared to using the uniform surface.
Okay, yeah. Thanks. Yeah, actually, for chapter 3, you indeed now mentioned the size, but you mentioned the size of the probe region. I was actually referring to the size of the inducers. So do you think that can also help to maximize your effect?
Yes, I think our stimuli is like either square. So in the psychophysics in chapter 3, so we also changed the square of the probe and also the shape. But when you change the size of the probe, you also at the same time change the size of the inducers. So far from our own data set, I didn't see a significant difference for the inducers. But I remember there's one paper, they used a much larger stimuli, so they have like a 35 by 24 degree of inducers. Their probe is just 9 degree. It's already bigger than our entire display.
Yeah.
So they do find a size when you do move to the edge. They do see the influence of the illusion. But I think for our own experimental setup, we did not see the effect.
Okay, thank you.
Okay, so I will now ask Professor Urdag if he wanted to ask one more question, as he was interrupted in his discussion.
Yeah, I think the layer probably we can skip for now. The layer question, I think I can communicate to you offline with that. So the question would be, you did surface-based analysis. Given that all these ROIs are relatively distinct from each other, do you think it would be a good idea to do actually not surface-based analysis, but directly on the voxel level? Because surface-based analysis has some strengths, some advantages, but it goes also with some problems with like anatomical misregistration and so on. So do you think it would be beneficial to do it actually on the voxel level, and how would you do that?
Highly esteemed opponent, thanks for your questions. I think it would be possible to do it on voxel level, maybe not have the population receptive field, but just have a normal retinopathy. Then you have the field range to give the location of the region where you want to be, and then you pick the highest activated voxels for each range. So I think this might be help to do it in a voxel level. But I think one good advantages of using our method is that you can more or less freely to pick the size of the probing region or in inducers, and also explore whether their effects on the illusion or on the cortical response.
Thank you very much. I think given the time, I will yield, and then if there's more time, I can ask more questions later on.
Okay, so the opposition will now be continued by Dr. Vincent van de Ven from the Cognitive Neuroscience Department at Maastricht University. And the floor is yours.
Thank you. Dear candidate, I also enjoyed reading your thesis a lot. I'm impressed with the amount of additional work you've put into...
Please use a microphone.
I'm doing it now. Great that you are putting extra work into your research, especially after you find partial or perhaps not none of the things that you're looking for, where you could just stop and say it's a null finding. You actually push through and try to find out where this comes from. And that is always a commendable thing to do, and in that sense, a strong example of how to conduct science in these new fields. I have a number of questions also about chapter 3, the brightness illusion, but I would like to start with 2 of your propositions that you stated. Proposition 5, early visual cortex may well engage in the processing of surface brightness. However, we could not find convincing evidence using 70 fMRI. And especially the first part, I think, is a good starting point, because it's also based in part on work that I did, where this was sort of in line with the statement. But then at the very final proposition, you state what gets us into trouble is not what we don't know, it's what we know for sure that ain't so. And I found that interesting because chapter 3 reads very much like a scenario for which you must be able to find brightness illusion correlate in early visual cortex, replicating work that's out there. But the work that is out there is actually quite variable. So some studies find some correlates in V2, and there's also a few studies that very adamantly claim that there's nothing going on in V1, V2, or V3. So I was wondering what you would think of your evidence if you actually approach it from the point of view of the alternative model, the symbolic representation. You actually mention it in your chapter, but then in the discussion you actually say, well, there's a lack of evidence for the isomorphic representation, but that doesn't mean that there's any evidence in favor of the alternative model. So I was wondering what you thought of if you approach it from this symbolic representation, how you would then interpret your data, and what you would think you would need in order to conduct a future experiment using this well-elaborate setup to see if indeed there is more evidence in favor of the symbolic representation.
Esteemed opponent, thanks for your compliments and question. The question is what can I do if I do the experiment from a symbolic theory view?
Yes, let me perhaps clarify a little bit. The entire chapter is written with the sense that you'll find something there. And the interpretation of the findings is indeed that there is a little bit of evidence of a probing type of effect when the luminance of the inducer goes down. And then there's a little bit of an increase in the probing region at the very start. But all is very, like the effects are weak and small, and there doesn't seem to be anything going on when the luminance increases. So you could phrase it as the glass is half full. There's a little bit of evidence, and if we would frame the stimulus a little bit better, we might find it. But it might also mean that what we're seeing is perhaps a little bit of noise on top of a proper lack of effect, because there is no neurocorrelate of brightness in V1 and V2. So if you approach it from that way, how would you view your evidence, and what would ideally be your next steps in pursuing whether indeed your effects are related to the symbolic representation rather than an isomorphic representation?
Thanks. I think because in the field there's already like a discussion whether it is involved or not involved. So we start the story by telling something it's very possible to be involved, but there's also plenty of studies either in fMRI or in other physiology studies. They didn't find the thing what they want. And I think from this point of view, I think the good thing of our results is you cannot fully conclude that we didn't find anything, because it does show some little bit of trend, like the antiphase relationship might be there, but our results is not in V2, so it kind of it's the sum of the all visual cortex, so V1, V2, and V3. So in this sense, we cannot say which region is really involved. But if you conclude, say, oh, we did find something, but we can also not say that, because especially the antiphase relation, the anti is very small. So it's not very significantly from 0, but it's almost. But if we have more participants or more data set, or if we have some a bit stronger illusion, we might be able to find it. So I think I would still say it's like a open question for future studies to keep trying.
Okay, fair enough. Thank you. You've used mostly fMRI and psychophysics in this case, but one thing perhaps you could think of are other methods like EEG or brain stimulation. Has this crossed your mind at some point to use other methodologies to see whether indeed lower visual areas are relevant in perceiving the brightness illusion?
Thanks for your question. I think if we want to, one thing I'm thinking about is like you can use TMS. If you do think this region is evolved, and we can disturb the function in this region, whether we can see whether the participant still can have look, can still see the illusion, or whether the illusion will be largely reduced through this kind of intersection, and we might be able to prove it in the opposite way.
All right, thank you very much. I'm going to give the floor back to.
Yes, there is still some time, so I'm going to look at our external guests and ask whether anyone of you have additional questions you would like to ask. I for sure have some, so otherwise. Professor Rudak, you wanted to maybe ask an additional question, you said?
I can ask a question. So the nucleus basal minor chapter, you argue that perhaps the problem is spatial resolution and contrast. But if I think about it, in the big brain data, you had approximately 40 cubic millimeters, given then, say, 0.7 millimeter isotropic resolution of anatomical, you have approximately 120 voxels. It's not much, but still should be visible. So what do you attribute that it's not visible in vivo, given that you should actually have enough voxels? What do you think is the cause of it?
For your question, I think indeed, if we directly resample the big brain mask to the anatomical data at 0.7 or 0.8, we can still get quite amount of voxels left for the NBM mask. But our functional data is much lower, so it's at 1.25 millimeter isotropic. And then when I resample it to the functional data, there is only 12 voxels left. So, and because of the signal in the NBM, because it's in the middle of the brain, and the signal there is already weak, and it also have two big wings next to it. So, and also close to the those air interface and stuff. So it makes the signal very noisy. So imagine you have a very noisy data set, and you have only 12 voxels left, but you are not sure how do you place those 12 voxels into those kind of noisy data. So I think it's not a good idea to use those kind of data to continue investigate its function.
Okay, thanks. I think the time is almost over, so therefore I will send my questions to the supervisors separately. Thanks very much.
Okay. I think there is 1 or 2 minutes. I actually have one question myself. It's a curiosity more than anything else, and it's the fact that so you don't find, chapter 3, you don't find the effect when you are increasing the stimulus contrast in the inducer. And you actually decided that in order to maximize the effects, you would make these changes very big. So you go to white. Do you actually think that actually this didn't pay out? So it would have been better. So if I understand well, there may be an option that actually would have been better to not make it go to white. Am I correct? So would it be better in that case, maybe to not make it change so much, the contrast in the inducer when you go up? Yes, maybe you can give me a very short answer, so, and then...
Thanks for your question. I think going to white. So from dark to white, I think is to guarantee you have you do get a brightest illusion, and so before we didn't think about going to white, it will be a problem. So we directly go from gray to dark, gray to white, and if now I'm thinking about it, so maybe you are right. So maybe we should do is how to in our own frame in chapter 3, what can we do to make the illusion bigger, or make each brightness make each luminous bright and dark even bigger, not just directly using clouds, introducing a new feature to the illusion. I think, as I also said before, so if you're inducing more features, it also might involve more mechanism towards that.
Okay, I think that anyway we have been told that we're done, and this means that the time allotted to you to to present your thesis and to defend it is now up. The committee will now retire in order to discuss the quality of your thesis and your defense. I ask you and your party to wait here for our return, and we will come back once the deliberations are done.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. can't fly by now, don't waste all your time. Cause I'll go the extra mile.
哦，那个，啊。
Yawen Wang, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor De Werd is authorized to confer upon you this academic distinction in accordance with Dutch university customs, and I invite your supervisor to now take the floor.
Dear candidate, dear Yawen, before I can give you the diploma and call you doctor, I have to read this out to you and you have to answer honestly. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
Then I go on. By the authority vested in us by law and conformity with the decision of the committee here present, I hereby confirm upon you, Yawen Wang, the degree of doctor and grant you all the rights attached to it by custom and law. As evidence of this, I now present you this with the degree certificate signed by the rector, the secretary and the other members of the committee, and affixed with the official seal of Maastricht University. I will say only 2 words because the Mark and also the other members in the promotion team did much more work than I did. But I just want to take this opportunity to say that I'm very proud of you. You did extremely well today. It was a thesis that didn't get there very easily. So you got there. And you should be proud. Today you really showed also in your defense that you're a true scientist. So be happy and be proud. And now more words of praise by Mark Roberts.
Thanks. Yawen, congratulations. This was a really nice defense, really nicely presented. It's beautiful to see all of our work together put in a single story. Really nice work that you did. And a very nice overall project. Yeah, as Peter said, your project didn't work out exactly as we thought it would when we started. But I think, honestly, that's the case for all projects, right? Nothing happens the way you think it will. But I think in your project, really, when we started it, we said this would be a very difficult project. So we already knew this was a very high risk of not finding anything, of finding it just too difficult. But we said, well, if we find something, this will really be something, right? So we thought it would be a high risk, but maybe with the possibility of a high gain project. And we thought when we thought about this project, the 3 of us, who on earth is going to come and try and help us with this almost impossible task, right? And we met you online for an interview when we put this grant to the China scholarship thing. And yeah, we were immediately impressed because you had a very good background in research. You'd been working in Denmark. You had a passion for doing MRI research. And what really struck to me, what I remember, was that you had this great background in neuroscience, right? That was because you were sat in the library, and behind you was all of the neuroscience textbooks, which I think you didn't know where you were sat, but that was it. So that for us was enough, right? And you agreed to come and join this very difficult project. At the beginning, I think everything started to go well, right? So we had some nice data quite quickly on. What did I write here? Yeah, the first years we learned a lot. We discussed all our papers. We also, you and I together, we supervised the master's project. Do you remember this? And I remember you gave a similar slide as you presented here, but I suggested, well, this is how you should present the importance of contour segregation. And this is when I learned that you were very fearless in most cases, but presenting a picture full of snakes, this was no good for you. And that was always difficult for me because, I don't know if you know this, but in front of your office there is this picture of some guy fighting off huge snakes. But there we go, you didn't know this. There we are. Because I always thought, well, should I say congratulations that you come every day to face your fears, or should I not point it out so that you don't, right? So it was a difficult thing for me all these years. Yeah, anyway, so the first amount of time passed and you gathered some functional data, and I thought it looked great, right? Because we had some activity that was kind of in almost the right place in one of the participants. I thought it was great. But it wasn't enough, right? So you looked more at the data and you said, well, we just can't be sure what this is. We can't really be sure where this is coming from. And the more you looked at it, the more unsure of it you were. And at the same time, you were writing this review paper that I had asked you to write. And the more you got into the literature, you realized that this project which you had come to Maastricht for, it wasn't difficult, it really was impossible. And you had the data to prove it, right? That's a great thing to do, right? You put this work together into this paper, and I really am immensely proud of it, right? It's a really important piece of work. It says something very meaningful in the field, and it says, well, this project is impossible now, but in the future, it will be possible. And you show the way that we are going to go and do this eventually, maybe. And for the moment, it also says important things about how you can image these difficult structures. I think you learned a lot, we learned a lot about how to do this white matter stuff, which you show for really the first time. This is an important thing. Yeah, but this project that we'd like to do, not now. So what do we do? Well, we sat together and we thought, we've tried high risk, high reward. That didn't work. Let's do something nice, something easy. Will definitely give us nice results, right? We'll just replicate something we've already done. We know it should be there. We're going to use the best methods, lots of repetitions, the best data we can have. You know how to do the PRF mapping. It's going to be fine. Yeah, and well, we didn't find what we were looking for. But, I mean, that's the reason we did the experiment, right? We didn't know what we would find. We didn't know if the effect would be there or not, and it turns out it's not. It's fine. Actually, about half of the literature agrees with you. So now we have half of the literature represented in our group. It's good. And we learned a lot also from this project, right? So the future projects in our lab, they will follow up what you have taught us. Actually, one of the decisions was that we would always do some 3 Tesla scanning before we went on to the 7 Tesla, right? And now we have new people in our lab. Yeah, they are just doing the 7 Tesla scanning, so I don't know what happened there. Anyway, yeah, one of the papers that agrees with your results, right, that there's no nice response in early visual areas, it has a really nice section all about the Van de Ven study and how wrong they did everything. And one of the things they said is, well, the difficulty with this stimulus is that you change the total brightness of the stimulus between the 2 conditions. So you can't really discriminate brightness and luminance the way we would like to, right? So we thought, how can we approach this? How can we address this problem for the follow-up study? This was at the end of 2019, and we came up with a very nice set of studies. We had it all planned out in the lab. Participants were just arriving to do our psychophysics experiments, and then they stopped arriving, and we all went home for 2 years. And well, what can we do? We moved our experiments to online psychophysics. We learned a lot I mean, it's kind of similar to the 7T. It's a new method. It's got its own potential. It's very nice to get so many participants with no effort. It's also got challenges, as we found out, right? You can't just do the thing we would have done in the lab online, right? So we learned a lot from there. And at the same time, you spent more time on your MRI data. You had a lot of interesting ways to analyze this data, stuff which Peter and I couldn't really help you with because you wanted to go really deep into it. And this was really nice that you reached out and you found Shri, who came and helped you, and also Minye, who really gave a lot of support. And it's kind of a shame that you are not on the promoter list because you gave really a lot of support through those years. Yeah, so if I was to summarize your project, as you also nicely summarized, it's been very challenging. It's been very rewarding. Right? You have produced, I think, 2 very nice papers, one with us and one with other people, right, now published. Yeah, you produced a lot of good work. So I had a feeling there was a whole lot of more stuff in this last paragraph. It wasn't. I wish you really every success in what you go on to do. I think you have good options if you want to carry on in a scientific career, and I think you have good options if you want to go and find something else to do in your life. Congratulations on the work. Thank you very much for coming and helping us with this project. We couldn't have done it without you, or it could have gone a very different way without your serious approach to it. Thank you and congratulations.
Thanks.
Dear Dr. Wang, also on behalf of the Maastricht University, I congratulate you with the degree you have acquired. It's also the time for me to congratulate all the people here present and thank them for being here, also ones online and joining us from abroad with hours of difference and so on. I hereby declare this academic session closed, and I will from far. And then just a word of caution to everybody. You can go ahead and go to the rafter. We will stay here and take a few pictures here and then on the stairs, and then we will join you. Thank you very much.
Yeah, so my